Hello, my name is Madison and I'm currently a second-year biochemistry biophysics major here at Oregon State University. I'm currently working as a physics undergraduate research assistant for Dr. Bo Sun, and I've had the privilege of working in this lab for almost 2 years. A large part of the work that's being done here in the Sun Lab is cancer research, in particular studying how cancer spreads and manipulates its local environment through a physical lens. Biophysics is a field heavily focused on applying physical principles to biological systems, and the more we understand the mechanics and dynamics of how cancer manipulates its local environment, the more effective and efficient anti-cancer drugs can be developed. Within cancer research, it's very important to understand and quantify how cancer spreads throughout its environment. The best way to do this is typically by studying cancer cells within an actual cancer patient. However, as you can imagine, it's very difficult and unrealistic to study cancer within humans. Within the Sun Lab, we aim to simulate cancerous environments within an experimental setting by using live cancer cells, spheroids, and 3D gels. A spheroid is a 3D mass of cancer cells meant to replicate or simulate a tumor. These tumors or these spheroids can realign and invade their environment much like a malignant tumor would within the body. We then place these spheroids within a 3D gel consistent of collagen fibers. Collagen is a protein that actually makes up much of your body tissue and is easily an example of how we can replicate cancerous environments within an experimental environment. And by simulating this environment, we are paving the way to gaining a better physical understanding of how cancer spreads throughout the body. My project within the Sun Lab is to model multifocal cancer. Multifocal cancer is when you have two or more cancerous tumors growing within the same quadrant of the body, or on the contrary, unifocal cancer is going to be when you only have a singular tumor growing within one quadrant of the body. Although there's no concrete proof that multifocal cancer is more aggressive than unifocal cancer, there has been evidence that multifocal cancer can put patients at a higher risk for developing larger tumors faster, which can impact the speed and spread of cancer throughout the body. I modeled this multifocal cancer by receiving 2 cancerous spheroids within the 3D collagen gel, and then I can track this invasion and influence that the 2 spheroids have on each other through using live cell imaging, and this live cell imaging is done by using a confocal microscope. The confocal microscope is able to give us an understanding of not only the 2D scale of the cancerous environment as well as the 3D scale, and it obtains this 3D scale by taking multiple images at multiple heights throughout the sample. And because we're gaining this 3D model, it's going to allow us to have a much more accurate understanding of the physical environment of how cancer is spreading and influencing its environment. We can then use this data to send to our collaborators who are able to develop a mathematical model that will then begin to explain the biophysical processes that are at play. Cancer is an extremely dangerous disease, and according to the National Cancer Institute, in 2024, in the United States alone, 2 million people were estimated to be diagnosed with cancer. Although in this lab we may not be directly developing anti-cancer treatments or anti-cancer drugs, the information that we are gaining and the data we are gaining on the physical understanding of how cancer spreads and manipulates its environment will ultimately lead to the development of much more efficient and effective cancer treatments in the long run. The work that we're doing in this lab is all part of a larger stitched effort by researchers all around the world that are hopefully trying to find a cure for cancer. My time in the Sun Lab has been extremely enlightening and has allowed me to apply my academic skills to a real experimental environment. Not only am I learning the experimental process by doing physical experiments and data collection, but I'm also learning about scientific writing and the publication process. The skills that I'm learning within this lab are going to carry over with me to my future career and hopefully help me long after I've left OSU. And I'm extremely grateful and proud of the work I've been doing within this lab. Thank you for being here and thank you for watching this video. Go Beavs!